I'd like to thank the organizers for this opportunity to speak today. I understand I'm the only person between everyone else and lunch, so I'll try to be fast. But I was asked to talk about some additional aspects of dosimetry related to target alpha therapy. I'm a clinical medical physicist at the University of Arizona, primarily in radiation oncology, but I'm also working bridging the gap between that we have for theranostics, our theranostics center, and being able to do clinical dosimetry, starting with some of the lutetium-based therapies, then hopefully in the future, some of the alpha-based therapies. And I'm also an optical scientist, and a lot of my background is in hardware and detector development. So I'll talk about a system that I've been developing for some time that hopefully will be interesting and relates to some of these aspects of being able to quantify measurements and tied to autoradiography. Quick disclaimer, the technology I've developed, I've founded a company that's commercialized it. And to start off with, at the Professional Society of American Physicists, American Association of Physicists in Medicine, their efforts in radiopharmaceutical therapy with alpha emitters. There's a committee that the chair is Robert Hobbs at Johns Hopkins, who's done an amazing job of working to form task groups, working groups, to be able to disseminate methodologies, the dosimetry, the science, and practice, and take an active role in education and implementing clinical practice cases. And I'm working on a working group that's under review that's for radiopharmaceutical therapy with alpha emitters. And some of the main goals of this is to review preclinical dose estimation techniques and also clinical aspects of it, but looking at imaging-based dose assessments, SPECT imaging, and also direct and surrogate isotopes, how those can be handled. And so we're just at the early stage of this, is under review, but we'll be co-chairing this with Dennis Bergeron here. And I think it's very important that establishing a lot of these standards and have a way that the community can easily access articles and methods to implement those. So a primary topic will be quantitative digital autoradiography and its growing role that we're seeing in radiopharmaceutical therapy. I'll highlight what's called the IKID detector. There are other autoradiography systems out there. Tom Beck here has invented one called the Alpha camera several years ago. It's AI4R, another company. But I think these technologies are very important in being able to provide a link between what's done preclinically up through the clinic. A little bit of background on the IKID camera. It stands for the Ionizing Radiation Quantum Imaging Detector. It was originally developed as a SPECT-based system that has very high spatial resolution, but it's sensitive to a broad range of radiation from gamma rays, alpha particles, betas, neutrons, and fission fragments. Here's a system that was built originally for preclinical SPECT, had 20 of these detectors, and was able to get very high spatial resolution on the order of about 0.35 millimeters. And here's an example of a system where you have a traditional SPECT system, and this is what you could get with the this IKID-based SPECT system. Here's an image of the detector when we're using it for autoradiography. It involves placing a sample on the detector. It uses a light amplification system that allows you to visualize up to individual single optical photons, and in real time allows you to generate an image. Stepping back a little bit about the technology, first time we really saw alpha particles was through the development device called the spinthariscope by Sir William Crookes. If you had a scintillator, in this case a zinc sulfide, and you had a radium source, and you put a lens there, and you went into a room that you would dark adapt to, you could see individual flashes of light, which were the individual alpha particles. And here's a more modern version of the similar principle, but this is using with the IKID, where you have an alpha particle interacting in a scintillator, produces a flash of light that gets amplified, and then that gets imaged onto a very fast CCD camera, multi-megapixel cameras. The setup involves taking sections of tissue, placing them on the scintillator, and then arranging them on the detector, or also using a microscope slide. And then the output is an image of the distribution of the tracer. The detector works by taking individual images from the camera. In this case, this magnified view, you see 3 alpha particles. And then in real time, it localizes each event shown here. In this case, the alphas appear to be a 2-dimensional Gaussian-like structure. And then you can take the individual pixels associated with this event and localize them to a single pixel or a single subpixel unit. When you do that, you can get very high spatial resolution, and you remove a lot of blur that you'd otherwise get with traditional autoradiography, like phosphorimaging plates. So here's an example with a tungsten foil that had a source placed on it. If you were just to add up all the light from all of the events, you have that light spread. You can remove that and count each event, and you get a counts image shown here, where you have much higher spatial resolution. And to further illustrate the point, here's an example of a digital autoradiograph. This was taken with astatine-211, looking at a lymph node section. This is in 1 second, you see there's 5 alpha particles, and then in real time, you start building up a counts image. And what's really powerful about this technique, if you're counting each individual event, is you can go back, you tag both the time and the location of each event. You can extract those events, and then you can plot the frequency of events as a function of time. And what you're seeing here is an exponential decay associated with astatine-211. You can then go back and quantify what that activity was at that time point of the biopsy or the necropsy that occurred. And here's an example of a biodistribution study. This is looking at a kidney, and after the detector is calibrated using a source that has a traceability measurement, you can quantify how much the sensitivity of the detector to alphas, and then you can quantify it. In this case, this was around 31 becquerels for this kidney section. This is the same study at a different time point, and here you have small regions of activity, relatively high compared to the normal, and then a lower activity region, but you can begin to start getting towards millibecquerel levels of activity you can quantify for astatine-211, which has a 7-hour half-life. It also works well with other alpha emitters. Here's an image of lead-212. This is a droplet that was placed on the scintillator and then imaged. And this was set to detect alpha particles. And what you're seeing here is the ingrowth of the bismuth-211 and then the exponential decay that occurs. Here's an image taken again with the lymph node section. And the point to this is you can have regions, I think it becomes very important to quantify these with the goal to eventually do dosimetry at this small scale. And there's regions of activity in this lymph node that are 20 times higher than the average background. And so it becomes important then to combine that with histology to look at the spatial distribution and be able to assess what the activity is. Here's an image with bismuth-213 with a 45-minute half-life. It also works with beta emitters. Here's the detector operating with samples from a yttrium-90 and then also lutetium-177 beta. And here's a recent section looking at lutetium-177 in a mouse kidney. And what you're seeing here is very high uptake and very small regions of uptake of the lutetium within the cortex of the kidney. The device, I think, it becomes a tool for also looking at source QA. This is a study looking at, in collaboration with colleagues at CDA, Stephanie Lamar, that we looked at electroplated disc sources that were used for radiation experiments to irradiate cells. This was, I believe, it was a plutonium-238 source, and it was a way to assess what the uniformity of the sources, where there are experiments being done that assume that the distribution of the source across the entire disk, the plated source, was uniform. But we have some regions where there's 5 times higher activity that are within it. So it's, for a lot of these sources, it can be a way to QA it. This was a recent publication from work at Matthias Sevenwa in Brussels at VUB, where it was looking at polonium-210 production within a bismuth target and assessing both at 2 different energies and seeing what the distribution of it was within the bismuth target. And There's also the ability to take and use it for source QA. In this case, this was a painted source that was placed on the detector. It was a plutonium-238 for radiation experiments. And then combining that with microcollimators, you can image very, very high activity sources. And even within the use of digital autoradiography, it's important to notice that your sample preparation is key. Here's taken in this first image with the source placed on a phosphor screen. The source was not completely flat, so it had some space between it. And so if you were to make assumptions that the uniformity of your source in A and B, it looks quite uniform. But when you start to make sure it's completely flat, you're using a collimator, you can have regions that have 100 times higher activity distribution, and it's very heterogeneously distributed. As mentioned, radiation oncology, we take a lot of effort to make sure that we're able to have sources that come from us, come to our clinic, and use a well chamber to calibrate those. What can be a challenge, though, is when you have sources made that you cannot directly interrogate. So in the case of, this is called an eye plaque. These are brachytherapy seeds that were, they're inside of a sealed packaging for sterilization purposes. You can't directly interrogate them. Imaging them is a challenge, but if you combine scintigraphy, in this case, high resolution scintigraphy, you're seeing individual gamma rays. This was an I-125 source, and you can use that to then quantify what the activity is in the source, and it's a way to, before it's placed in the patient, be able to assess that. So here's an image after a 5-minute counting of those sources. And we're also working to extend that now to other cases. There's alpha emitters that are diffusion-based ones, a company called Alpha Tau, and they have an alpha darts, which is a seed, a brachytherapy seed that's implanted in tumors. And here's a method that you can place it in there, image it, and see what the diffusion is within that, but also looking at possibly quantifying the seed activity as well. For dosimetry, there's a nice paper that George Skouros published back in 2020 that talked about some of the challenges and processes. And his conclusion was that the scale which we do calculations, they need to arrive at absorbed dose estimates that are more likely to predict what the biological effect was. So that what comes into play is looking at dose estimates starting from the macro and the micro, and looking at it the whole organ level, the sub-organ level. And so there's a need to be able to link these two. And autoradiography plays a key aspect of that. And now there are tools that will allow you to be able to quantify that. This is a technique also at Gothenburg. Tom Beck had worked on a similar aspect of this, where you can do small scale dosimetry, where you need to sample, do quantitative autoradiography, but include enough samples where you can look at the contribution from an alpha emitter some distance away from a slice of interest. That's done by taking and sectioning tissues at different thicknesses, consecutive tissue sections. You image them. This is a case from a lymph node biopsy in collaboration with Fred Hutchinson Cancer Research Center in Seattle. The process involves taking the tissue sections, registering them, which by itself can be a very tedious process. You have many tissue sections, make sure they're aligned. Having a voxel-based dose kernel, this is for astatine-211, and voxelizing that to match what the positive energy is for the, on average for astatine-211, convolving that with your image, and then you get a dose rate image, but units are now in the micrograde per second. This is looking at 2 time points, and this is a dose rate image. What can be a challenge with the alpha emitters is that it can be very heterogeneously distributed. So taking care to localize low activity and high activity areas, assessing those regions, combining them with histology, and then doing this over multiple experiments to then integrate and get a time activity curve, integrate that to get the estimated dose to those regions of high activity and low activity. It's a challenging process, especially having so many tissue sections, being able to do that. So we did a study in collaboration with a group at UCSF, where we looked at the number of tissue sections needed to do the dosimetry. And we found that if we were to clone the tissue sections, making some assumptions that the morphology didn't change much. That can vary, so it may not be practical in all cases. But in some cases, you can start to get under estimates of within 10% using a few tissue sections and applying this dosimetry estimate. There's also work with looking at parent-daughter isotopes, imaging them and quantifying them. We talked a lot about actinium-225, and the concern many times is that the unbound parent or daughter isotopes, they can relocate to normal organs and tissues. And so George Sikoros kindly presented some slides where, and others here at Germany were showing that you can do quantitative SPECT CT. This is looking at the francium-221 and bismuth-213. And this is at another time point, but showing that you can start to do quantitative SPECT CT. There's still the challenge that remains, though, in terms of you have these images, but what does it really look like as you're approaching the microscale level? And so we've been looking at taking the temporal and spatial information that you get from technologies like these and identifying, in this case, this is looking at quantifying the actinium-225. So here I'll note there's a francium-221. It has a 30 that decays after alpha emission to astatine-217 with the 32 millisecond half-life. And so you have two alphas back to back. And so we did a study, this is in collaboration with researchers at Cornell, where we looked at generating all of the events that were taken. This is within a kidney section of a mouse, and at different time points, looking for one alpha, and then some at roughly the same space, some time afterwards, you get another alpha decay, and then repeating that many, many times. When you do that, you can then start to form images. In this case, here's the francium-221, and then looking at the images on this side, this will be the astatine-217. And to initially demonstrate that, we took all the events and binned in time all of the events with the delta T's and generated this curve here where estimated half-life was about 33.5 milliseconds versus what was published around 32 milliseconds on the NNDC. So here you have an astatine-217 image. This was an experiment in collaboration with Anders Jofsen at UPMC in Pittsburgh. And this was actually looking at how much francium is at a given time point. This is a very challenging measurement. The half-life of francium is only around 5 minutes. And so here we did an experiment where we harvested a mouse kidney, sectioned it in half, placed it on the detector, and then applied that technique and generated an image where it's there. In this case, there's a few counts. There's a first pass at it, but here you're seeing the histogram of the counts as a function of time with the exponential decay of francium within that mouse kidney. So the summary, kind of the big picture in this aspect is the role of quantitative digital autoradiography. It allows you to begin to start assessing the parent and daughter isotopes. It's important to be able to decay correct and do it in a way that you have an accurate measurement, generating dose estimate, dose rate images. You have to repeat for multiple time points, and then ultimately you need to combine with histology, assess histology, where is it going, what are the cell types. Immunohistochemistry will probably be an important role as well. And the one final area we've also recently been working on is assessing cases where what if you co-inject alpha and beta emitters. This is recent publication showing that care needs to be taken with each vector, each ligand, in terms of estimating where it's going to go, not just assuming that it's going to co-localize. It needs to be studied for each ligand. This was looking at a gallium-68 DOTA, where they varied the charge, and they showed that you get different distributions based on the influence of the net charge. We started looking at this with actinium-225, lutetium-177, which could be co-injected as both a combined therapeutic. This is in collaboration with Sarah Chill at Cornell, and the goal was to look at with the detector, you can image both alphas and betas simultaneously. You can then generate them and parse out images that will allow you to see both simultaneously. So this was looking at, in this first column, these are total counts. The next column is the alpha counts. And then the last window is looking at just the betas that were detected. And so we had lutetium only as a control, actinium only as a control, and then the mixed isotopes in these last two rows. And then here in this red and green, looking at where green is the beta window and alpha is the red window. And so what's really interesting about this is a bit surprising. This is a ligand. There were some slight modifications to the ligand, but the assumption was that they would likely co-localize. And when we image these, this was looking at the mixed tumor. Shown here, you'll see regions where there's clearly very little or no alphas have gone there. We have other regions where the actinium has been absorbed. So we're still not totally sure what the mechanism is going on. We've looked at histology, trying to better understand. The picture isn't quite clear, but it definitely opens up the case where you could make an assumption where if you injected this, maybe your alpha emitter, the range is so short, it may not. You may have areas where you miss. And so the idea is if you have betas, you could then come back, possibly target those areas. But I think it opens a whole space that it really needs to be looked at for every type of ligand where it's going to localize. This is within the kidney. So even the kidney, when this is the mixed isotope of lutetium and actinium-225. So we have your alpha counts here, and then you have on the right the beta. So we're even with this ligand, we're seeing different uptakes. This is another one with a tumor where the ligand was identical, and except for the difference was the lutetium and the actinium. So even with it appears with the alpha counts, you get pretty uniform distribution of the actinium. Here you have the beta counts where it's more localized, but you have regions more at the gross level where it's a different distribution. And then same thing with this other study where they matched, where the injection with alpha counts, it's more in the cortex, and then lutetium, some of these you have different lobes within it that are localizing. So there are a number of companion therapeutic alpha emitters that are being looked at. One through DOE is cerium-134, which is a PET tracer. So the idea that you could inject this PET tracer and see if it localizes. And did another study with the James Kelly Laboratory at Cornell. Similar experiment looking at actinium, in this case, the cerium-134, the positron emitter, and then a combined study of mixed ones. The ratio of these is about 50 to 1 for those. So again, looking at the alpha counts, you'll have regions where it's heterogeneously distributed, or there's concentrated regions. But when you look at the beta or the positron emitters, you definitely see a different uptake within those. So I think it starts to become more and more important to look at these and at the microscale level to not just assume that they're going to co-localize. This is looking at a tumor, the same study for the cerium-134. It seems to localize quite well. But again, this will be something that's needed to be looked at and quantified. And then this final image, this is looking at what that was interesting. This is a liver section from that study, the cerium-134, actinium-225. The alpha counts, the alpha is appears quite uniformly distributed, but you look at the cerium-134 and you have these regions of very high uptake, these focal spots that are, I don't have the values here, but maybe 10 times, 20 times higher than whatever the background is. So I think it's looking at things on the microscale is very important, and then to extrapolate those eventually towards human use, there's a lot of intermediate steps that need to happen. And I think this all goes back to being able to understand how well you can quantify your sources, what you're injecting, and starting this even at the preclinical level to make sure that the values you have and what your interpretations on these are accurate. And just in conclusion, I'd like to acknowledge all the great team of people I've been able to work with over the last several years. And thank you for your time.